Today, we're diving into an incredible detective story, one that took over 150 years to solve. It's the story of how we, as a species, went from having no idea how life worked to being able to read, and now even write, the book of life itself. It's the history of genetics, from humble beginnings to a future we're just starting to imagine.
So, here's our game plan. We're going to see how the story unfolds, starting with some hidden rules, connecting them to physical stuff in ourselves, decoding the molecule of life, cracking the code itself, learning to read the whole thing, and finally, editing it.
All right, Chapter 1, Mendel's Hidden Rules, the Mystery of Inheritance. I mean, this is a question that's been around forever, right? Why do kids look like their parents? How does that even work?
And this quote kind of sums it all up. For centuries, people just assumed inheritance was, well, messy, like mixing paints. You get a blend of your parents. But then, in the 1860s, you have this monk, Gregor Mendel, just quietly working away in his garden with pea plants. And what he found was that it wasn't messy at all. It was clean. It followed predictable mathematical rules, a total game changer. So he called these things factors. And the key was there were these distinct little units of heredity. They didn't blend together. They were passed on whole. The problem? He had no earthly idea what they were or where they were hiding in the cell. It was completely abstract. I love this analogy. It was like he figured out the rules of grammar for a language, but he'd never actually seen a single written letter. Okay, moving on to part 2, the chromosome connection, finding the physical evidence. So Mendel's work was amazing, but it was just a set of rules, right? A concept. Science needed something tangible. The big question became, what physical thing inside our cells is actually carrying these factors? And this is where it all starts to click. In the early 1900s, microscopes got way better, and scientists started seeing these little thread-like things inside cells when they were dividing. They named them chromosomes. And then came the aha moment. On the one hand, you had Mendel's abstract factors, just an idea. On the other, you had these physical chromosomes, and they were behaving in exactly the same way Mendel predicted his factors should behave. The abstract was becoming physical. And here's how they knew. The process is called meiosis. This is how egg and sperm cells are made. And it's just brilliant. The chromosomes pair up, line up in the middle, and then, boom, the pairs get pulled apart into 2 new cells. That's Mendel's law of segregation right there in action. And the way they line up is totally random, which explains his other big law, the law of independent assortment. This wasn't just some random cellular dance, it was the physical proof. Mendel's factors, which we now call genes, were sitting on these things called chromosomes. All right, section 3. Decoding the double helix. So we've got the pages of the book, the chromosomes. But what were the letters? What was the ink? What molecule could possibly be amazing enough to hold all that information and be copied over and over again? Well, the answer was a molecule called DNA, deoxyribonucleic acid. But it's not just floating around like spaghetti, it's packed incredibly efficiently. A long stretch of that DNA helix gets wrapped around a core of proteins, kind of like thread around a spool. That whole unit is called a nucleosome, and these nucleosomes line up like beads on a string, which then coil up tighter and tighter to form the chromosomes you were just talking about. It's an amazing storage system. And then, in 1953, came the breakthrough, the discovery of the double helix. I mean, look at it. It's beautiful. And its structure immediately solved 2 problems at once. The ladder-like shape is super stable for storing information long-term, but you can also just unzip it right down the middle to make a perfect copy. It was the perfect molecule for the job. So, Chapter 4, From Gene to Protein. We've cracked the code in a way. We know the letters are A, T, C, and G. But how do you get from a simple 4-letter alphabet to us, to all the complexity of a living thing? The secret is something called the central dogma. Think of it like this. Your DNA is the master blueprint, locked away safely in the nucleus. You don't want to damage it. So the cell makes a temporary copy of one section, one gene. That copy is called messenger RNA. That's transcription. Then this messenger RNA travels out to the cell's factory, a little machine called a ribosome. The ribosome reads the copy, and that's translation. And as it reads, it builds a protein, amino acid by amino acid. And proteins, they do everything. They're the bricks, the motors, the workers of the cell.
Okay, part 5, reading the genome. By the 70s, we were getting pretty good at reading a single gene. But that's like reading one word in a giant library. What about reading the entire book? The whole genetic instruction manual?
And this number shows you just how big a challenge that was. 3 billion letters. Not a million, a billion. 3 billion DNA base pairs in the human genome. It's not a short story. It's a massive, massive encyclopedia set. The scale is just mind-boggling. Yeah, how do you read a book that long? Seriously, think about it. If you try to read it out loud, one letter every second, without stopping to eat or sleep, it would take you almost 100 years. So how on earth could science ever hope to read it?
Well, the answer was a technological race. And this timeline is just wild. Back in 1977, the first methods came out. They were revolutionary, but super slow. You could only read tiny little bits at a time. Then, in 1990, the world launched the Human Genome Project, a massive international effort that took over a decade and billions of dollars. But look what happened after 2007. Next-generation sequencing came along, and suddenly, a process that took years could be done in hours. What was once a monumental achievement became a routine lab procedure. The pace of change is just incredible.
Okay, last chapter. Editing the book of life. For all this time, 150 years, we were just learning to read the code. That was the whole game. But there was always this other, bigger question looming out there. Could we ever learn to write? Could we become editors? The answer, amazingly, came from bacteria. It's a system called CRISPR-Cas9. And it's basically a souped-up immune system that bacteria use to fight off viruses. Scientists figured out how to harness it. You can think of it like a biological find and replace tool for your word processor. It's a set of molecular scissors that you can program to find and cut a specific DNA sequence with a precision that, honestly, sounds like science fiction. And how it works is just so elegant. It's a 3-step process. Find, cut, replace. First, you create a little guide RNA molecule. It's designed to match the exact DNA sequence you want to edit. It leads the Cas9 enzyme, the scissors, directly to that spot. Second, the enzyme cuts the DNA, a clean, double-stranded break. And third, the cell's own natural repair system kicks in to fix the break, and scientists can trick that system into adding, deleting, or replacing the genetic code at that spot. It's incredibly powerful. So, what does this power actually mean? Well, the potential is just staggering. We're talking about the ability to go in and fix the single-letter misspellings in our genetic code that cause horrible diseases. We could potentially correct the mutations for inherited conditions like sickle cell anemia, target the fusion genes that cause certain cancers like CML, or even treat complex mitochondrial disorders. This isn't just an academic exercise. This is a revolution in medicine. So you see the whole arc, right? From Mendel's quiet garden, wondering about his pea plants, to holding the power to literally edit our own DNA. It's an absolutely incredible journey. We've gone from being just readers of life's code to being its authors. And that, of course, brings up some really big, really profound questions for all of us to think about, which leaves us with the ultimate cliffhanger. We have the pen. What story should we write?